Hi everyone, again welcome to the structural biology course. It is the continuing module where we are discussing structural biology techniques I am talking about, and now we are continuing with the discussion of X-ray crystallography. I have talked about the basic, the background and the starting of protein crystallization. Now in this class we will start with the goal of protein crystallization. What is actually our goal? Definitely to obtain good crystals. So we will discuss what are good crystals in terms of science. Is it the crystals which look good are good? Is it the crystal which are actually good? We will look at them. Why getting good crystal is important, understand how crystals grow, techniques for crystallizing proteins and strategies for optimizing crystal growth. So, protein crystallization is a process which you could call a development of a thermodynamically closed complex closed system, where we develop it in such a way. If you look at, we have the protein solution and we have the reservoir solution. The reservoir is made of precipitant which actually push the protein towards precipitation, and when it goes through the change of phase from liquid protein solution to solid precipitation, there is a small unique phase point, phase juncture, where the protein crystallizes. So, what we do? We have precipitant, let us say this is one precipitant condition, 30% weight by volume polyethylene glycol 5000, 1 molar NaCl, 50 millimolar sodium acetate pH 4.5, and protein is 100 milligram per ml, 50 millimolar sodium acetate pH 4.5. Now, what we do? We take 2 microliter of the protein and 2 microliter of the precipitant and keep it on the glass side. You could imagine cover slips which use in biology as a glass slide, then we put grease there and then we put back it on the reservoir place where it produce the closed system, and then we check for air bubbles to ensure that this is a closed system. So, what actually happened now? If you see the reservoir solution, we put 2 microliter, the protein solution we put 2 microliter. So, the reservoir solution with respect to reservoir solution is now half concentration. So, here the reservoir is 30% PEG 5000 and 1 molar NaCl and 50 millimolar sodium acetate. Now, sodium acetate is present in both the cases. So, the 30% PEG here becoming 50% PEG, and that is why this is the less concentrated solution. So, water will come from here to there, that will help making slow dehydration. That is where the principle of crystallization should, and by setting that we could get large crystals in 15 minutes, in 1 month, in 1 year, in 2 years, 3 years. I personally got crystal after 1 year. So, how long you have to wait? No one knows. That is the problem of crystallization. Supersaturation is very important in terms of crystallization. What is supersaturation? To add more of a substance to a solution than can normally be dissolved. So, you know that much solid could be solubilized in a solution or in a solvent, you put more than that. So, it is called supersaturation. Supersaturation is thermodynamically unstable state. You could understand whatever is possible, you are putting more than that. You could eat 100 grams of rice and I am putting 500 gram in your plate and make you bound to eat it. So, after eating you will feel uneasy. So, same like here, it is the solvent is feeling uneasy and that is why it is a thermodynamically unstable state. This is achieved most often in protein crystallography by vapor diffusion or slow evaporation techniques. We will look at that. As I told, you have the drop where the protein and precipitants are there and water would be coming from there, because the reservoir condition is half here. Zones are extremely important for protein crystallization. Zone 1 is metastable zone. This solution may not nucleate for a long time, but this zone will sustain growth. It is frequently necessary to add a seed crystal. So, after you set up the crystal drops, you have to keep checking under microscope. If you see clear zone, this is the zone 1. Zone 2, nucleation zone, protein crystals nucleate and grow. Zone 3, precipitation zone, protein do not nucleate, but precipitate out of solution. So, if you are lucky, you will get zone 2. If you get zone 1 and that remains there, after some time you have to start thinking different strategies like increasing concentration of the protein, increasing concentration of the precipitant and anything. If it goes to zone 3, then immediately you know that this condition is not fit for crystallization. You have to again think for reducing everything. Nucleation, as I told, is a very important term. The phenomenon whereby a nucleus such as a dust particle, a tiny seed crystal, or commonly in protein crystallography a small protein aggregate starts a crystallization process. Remember, I talked about what you are doing. You have a protein solution, you add precipitant, there is slow dehydration. So, the concentration of protein is increasing on the drop, and slowly through this dehydration there is a change of phase from liquid to solid. In between there is a one unique phase point where the crystal would grow, and you are very lucky if you get that. Common challenges: if the supersaturation is too high, too many nuclei form, hence an overburdens of tiny crystals. You get tiny crystals, you get crystals, but these crystals are not possible to diffract. So, you cannot proceed from there. In supersaturated solution that do not experience spontaneous nucleation, crystal growth often only occurs in the presence of added nuclei. Sometime it have a potential, but it needs few crystals, tiny few crystals. You cannot add a full grown crystal and you get other crystals. You have to put a micro crystal. How to get micro crystal? There are many techniques. One which people commonly use is seeding. So, we could do seeding using tiny crystals. Protein crystallization, cessation of growth caused by development of growth defects or the approach of the solution to equilibrium. Mother liquor, the solution in which the crystal exists, this is often not the same as the original crystallization screening solution, but is instead the solution that exists after some degree of vapor diffusion equilibration through dialysis or evaporation. What this is talking about, when you set up a crystal, it is called mother liquor, right. What you have, if you have some concentration of the precipitant, it would become half, but because it is half and because you develop a thermodynamically closed system, what happen? Water goes from the drop to the reservoir. So, slowly the concentration of the protein as well as the precipitant would keep increasing in the drop. So, the drop which you think the mother liquor, you calculate the concentration where you initially started it is changed now because of the process of dehydration. Unit cell in a crystal. Unit cell is very important. A unit cell is the smallest repeating portion of a crystal lattice. Remember, I talked about crystal. Crystal is an array of things, but suppose it is a array of pens. In one unit cell there might be 1, 2, 4 pens. It is not necessary that in the unit cell the number of pens, in the case of protein crystal the number of protein should be 1, it might be more than 1. It could also be considered as the basic building block of the crystal. As like we know, one cell, million of cells organize the body. Similarly, one unit cell, million of unit cell, if you see this is one unit cell and these are many unit cells, they build up the crystal. The whole crystal can be generated by repeated unit translation of the cell in 3 dimension. And here in unit cell it is characterized by its axis A, B, C and angle alpha, beta, gamma, as you could see here. Obtaining protein crystal. Obtaining suitable single crystal is the least understood step in X-ray structural analysis of a protein. See, you just in the history in the last class I told this process is continuing for more than 180 years. It is recorded for 180 years. So, process of crystallization is going through long time, but still now we did not understand the science behind. Protein crystallization is merely a trial and error procedure in which the protein is slowly precipitated from its solution. You just do your work best, you do the target selection, you do the optimization, you do the host choice, you do the cloning, you do the transformation, you do the overexpression, you do the purification, get the protein, set the crystal, then you go home, every day come check and just hope that one sunny day you will find a crystal. That is the part we crystallographers hate about crystallography. And people say crystallization is an art, crystallography is a science, crystallization is an art. So, what are the important factors which can affect the formation of protein crystals? We do not understand the science behind, but we could have studied more in depth and find out the reasons why. Factors that affect crystallization. Purity of protein. Purity of protein is one of the most important and critical thing to get a crystal. You know, when we used to do kinetics or spectroscopy experiments, we do not care if our protein is 90% or 95% pure. But when it comes to crystallization, we try our best. We at least go for 2 rounds of purification. We do nickel NTA followed by size exclusion, and before every crystallization set you are going to set up, it is advisable that you go through one size exclusion based purification, because that would make your protein confirm freshly going through the beads and folded properly. If your protein, when you are freezing it, there would be some portion of the protein denatured, even a tiny portion, that could prevent your crystallization. So, budding crystallographers, this is the advice from me, when you are setting up new crystals, before that always go for a round of size exclusion purification, even if you have an ultra pure protein. Protein concentration, you have to optimize protein concentration, and there is no parameter to optimize. So, you have to do trial and error, but if you keep high concentration, there is a good chance you understand it quickly. Starting condition, makeup of the protein solution, how you make the complex, when you are setting up enzyme which is like, suppose the enzyme is taking care of A plus B to C plus D, you will always pick up A and D, B and C. you never take A and B, C and D, because when you take A and B, your enzyme will convert it to C and D, and then if this is your enzyme, in one side there would be A and C, in other side it would be B and D. So, this would be heterogeneous condition, and heterogeneous condition never support crystallization. Precipitating agent, no one could say that. There are companies which have developed a lot of research, developed a lot of follow ups with condition where they get crystals and then they make their like sample screening samples like that, but nothing is known to be working for everything. There is no rule. So, you have to do more and more, and you have to do optimization. Temperature play a lot of role. So, this is advisable that when you are setting your crystal for the first time, you set it in at least in 4 degrees, 12 degrees, 20 degrees and 37 degrees. pH, you have to make screening with different pH. It is better that you know where your protein is stable, in which pH. Additives, detergents, reducing agents, substrates, cofactors, and if you look at this, in one word, this is shouting that with all our experience of 180 years, we have no idea who are the important factor that affect crystallization. It is depending on your protein, you have to find it out. And homogeneity in the crystallization solution, which I have already talked about. Now, I will talk about a very interesting consortium. It is called Structural Genomics Research Consortium. I talked about next generation sequencing. I talked about this is the technology which changed the spectrum how biology is looking at. After the success of next generation sequencing, where they have done human genome sequencing, and through human genome sequencing they have like got the collaboration between good universities like MIT, Harvard, Stanford, Caltech with big companies like Applied Biosystem, Illumina. They are doing high throughput gene sequencing projects. The high throughput gene sequencing projects are providing them millions of genes, and that means they have millions of protein sequence, but to proceed further means to go for biomedical problems, to go for disease identification, to go for drug development, they need more structures, more folded proteins, and there is a huge gap I have already discussed. To fill up that huge gap, there was a development of an initiative by Protein Structure Initiative of National Institute of Health US called Structural Genomics Consortium to determine the 3-dimensional structure of medicinally, health related and biotechnologically important proteins. So, by making a consortium, they got success that now they could sequence any genome. So, they are thinking their initiative is to develop further consortium which would solve high throughput way any structures, especially which are important. So, this consortium aims to places all its research output in the public domain without restriction and does not file for patents and continues to promote open science. These have several groups: New York Structural Genomics Research Group, Northeast Structural Genomics Center, Midwest Center for Structural Genomics, Joint Center of Structural Genomics, and CSMP and GPCRs are two initiatives. You know GPCR, that is G protein coupled receptor, and CSMP is the consortium for structure of metalloproteins. These are two rare consortium where they are trying to develop a high throughput initiative for membrane proteins which are very neglected. So, I had opportunity to work with this Midwest Center for Structural Genomics while I was in my PhD in University of Illinois. Northwestern University, they have house this facility mainly. So, we used to go there and work on the facility. I have further opportunity to work on New York Structural Genomics Research Consortium. This is led by Steve Elmo. My lab, where my PI was John Blanchard, he was having a neighboring lab to Steve Elmo. So, we had the opportunity to work there, to sit down, and we have seen how mammoth work they have built up. So, what was the goal of Structural Genomic Research Consortium is classifying all available genomic sequences to establish a prioritized target set. So, they want to pick up the way I told you select genes. So, they want to select like a lot of genomes instead of genes, cloning, overexpression, for purification at a high throughput way, purifying and crystallizing native and derivatized protein for X-ray crystallography, collecting data and determine structures, analyzing structure for fold and function assignment and homology modeling of related proteins. So, they know that when they are doing high throughput, they get some proteins crystal which they are not getting, they get all other proteins. So, they understand if there is fold matching, they also solve the structure of these proteins by computational techniques. Here I am representing a very interesting scenario. I hope you guys would give proper attention there, because this will tell you a lot of things. I am presenting the data from Joint Center for Structural Genomics, which was established at 2000, and this is the data after 10 years 2010. Selected target of that consortium was 33,209. They have cloned 27,959 proteins. They have overexpressed 27,640 proteins. They have crystallized 2,128 proteins. They have solved the structure of 1045 proteins, and they have deposited in PDB 968. If you look at the percentage, from selected target to cloning, they have a success of 84%. From cloning to overexpression, they have 99% success. From overexpression to crystallization, they have 8% success. From crystallization to solution, they have 49% success, and from solution to deposition to PDB, they have 93% success. So, you could clearly see which is the blocking point, and that is why from the introduction I am giving so much importance to the process of crystallization. The whole consortium is actually being closed because they think that the idea they had, high throughput, would not be possible. They need personalized care to do some surface engineering and different strategies which I am talking about, and that is the only way to get success. So, what was the realization here? When you are working with gene, you have a high throughput process, you could sequence all the genes, but from knowing the sequence, cloning, you probably have good success, but from protein to structure, a big problem is this crystallization. So, growing crystal is clearly a hurdle, but why it is such a big hurdle? I would try to give you an explanation in terms of thermodynamics. But before that, I would like to show you the thing I talked in the last class, why single molecule is not good enough to get diffraction. So, in a crystal, the diffraction signal is amplified by the large number of repeating units, right. When you see diffraction from a single molecule is actually not currently possible to measure, whereas when you have a 100 millimeter cube crystal, it contains 10 to the power 12 unit cells. So, it is much more bigger, mammoth, and have a architecture where the diffraction intensity is much higher, because diffraction intensity is proportional to the number of unit cell in the crystal, which is the formula by Darwin in 1914. In a crystal, the ordered periodic arrangement of the molecule produces constructive interference. I am sure you guys have knowledge of 10+2 level, where you know about a wave. When a wave comes and when two waves work together, there might be development of constructive phase and destructive phase. It should be constructive to get it. So, if you look at the pattern closely, you will see that in all the direction how they have developed the architecture of a 3-dimensional crystal. When a crystal is ordered, strong diffraction results from constructive interference of photons. You see the incident X-ray which hit the crystal. Interference is constructive because path lengths differ by some integral multiple of the wavelength. What I am talking about is a mathematical term which is called 2d sin theta equal to n lambda, but I am not talking about the derivation of the term. I will show you the derivation at the later part, okay. But this is, you could have understand that this is the famous Bragg's law we will talk about. So, when this situation is possible only because the diffracting objects are periodic. To be periodic, what have to happen? Again think about the array of panes. All your panes have to be in equal distance, then it would be getting the periodic function, and then it would be in phase. So, detector will get that and you will get good signal. So, but if this is not periodic, if there is irregularity, you will see that the crystal is not giving good diffraction. So, irregularity in orientation or translation limits the order and usefulness of a crystal. So, if you look at this crystal, this is perfect. This you see that there is rotational disorder, this should be here, this should be here. So, rotational it is wrong, and here you see this is having translational disorder. So, this is also wrong. When you have them, the disorder destroys the periodicity leading to streaky, weak, fuzzy diffraction. So, now you know you have to get a crystal, but you also have to get the crystal in perfect order. What is the effect? If it is perfect order, you see that is the diffraction pattern. This is what the data we collect, we will talk about that. Here when there is a rotational problem, you see that you do not get the spots properly. Here you get the spots, but if you look at, you get the mix up of the spots. So, this is also not good. So, they are wrong actually. So, it is necessary that you get a crystal, but the crystal have to be in perfect order. Rotational disorder and translational disorder. Disorder destroy the periodicity leading to streaky, weak, fuzzy diffraction. So, what makes crystallization such a difficult challenge? If we look at the thermodynamics, we know that del G equal to del H minus T del S, that is the thermodynamic formula of anything happening spontaneously. So, for crystal growth, del G crystal growth equal to del H crystal growth minus T del S protein in terms of crystal growth, and del S solvent. So, this del S protein and solvent is the entropic term and del H crystal is the enthalpic term. Do you understand what is enthalpic and what is entropic? If you remember the classes where I talked about how the protein interact non-covalently, you will remember I talked about hydrogen bonds. Presence of hydrogen bond includes the enthalpic term. What is entropic term? If you look at, there are proteins and there are solvents. When a protein grows, number of amino acids come together to form protein. Let us say 250 amino acid form one protein. So, formation of protein is entropically unfavorable. How you understand entropy very easily? Let us say you have a stadium. Okay. When you come to the stadium, you will not get response from the audience. So, you cannot enhance the noise of the stadium. So, you cannot enhance the entropy. Now, if Salman Khan comes here, the whole stadium would be erupted. So, Salman Khan develop entropically favorable situation in comparison to an Aam Aadmi like you or me. So, that is what entropy is. Entropy is increasing noise, entropy is disorderness. Okay. So, this is the representation of protein in solution and this is the ordered representation of protein in crystal. The first question is del H crystal favorable? The answer is yes, del H crystal is modestly favorable. Statistics says 0 to -17 kilocalorie per mole, because if you see in protein in solution there is no interaction, whereas in protein crystal there are lattice contacts in large area, it is specific and the interaction makes it rigid. So, this is favorable. Is T del S protein favorable? If you look at protein in solution and protein in crystal, you will see that protein in solution is actually could move freely, whereas protein in crystal could not move freely. So, here 3 degrees of freedom in orientation and 3 degrees of freedom in translation, whereas here 0 degrees of freedom in orientation and 0 degrees of freedom in translation. So, this is currently this is Salman Khan and this is you. So, this is entropically, the formation of crystal is entropically unfavorable. So, when it is unfavorable, it is plus 7 to plus 25 kilocalorie per mole. Next question is T del S solvent favorable in time of crystal growth? This is the protein in solution and this is the protein in crystal. Now, you see, in case of protein in solution, though the protein is moving, but the water molecules are attached with the protein. So, the water molecules are moving, but they do not have apparent movement. It is like you are moving in the car is going from one place to other, but you are not moving, right. So, the car is getting work, you are not doing anything, right. But here in protein crystal, proteins are not moving, so that allow the solvent to move freely, and that is why here in case entropically winning. So, for the solvent Here 0 degrees of freedom of orientation and 0 degrees of freedom in translation. In here it is 3 degrees of freedom in orientation and 3 degrees of freedom in translation. So, it is entropically favorable, minus 7.5 to minus 50 kilocalorie per mole. So, if we look at overall effect, del G crystal equal to del H crystal minus T del S protein plus del S solvent, del G crystal equal to negatively small favorable, large unfavorable and large favorable. So, overall crystal growth is in thermodynamically small favorable. So, that is why it is difficult, we get crystal, but there is a chance of around 8 to 10% if you do your other works perfectly. So, is there any other way to improve? Is like we could improve that chances, because you are thinking like I am going to choose my career to be a crystallographer, but if I do everything correct and then do not get crystals, what will happen? It is not actually that bad. Why? Because you could take strategies, personalized strategies to lessen the entropic penalty, the del S protein. How? You could eliminate floppy mobile termini. Like I talked about his tag, you could have cut the his tag, or you could have cut other part of the protein which is not contributing to the ordered structure. So, look at the protein, we have taken this part, and this protein, the full protein was not giving us crystal, whereas after we have taken out this part we got crystal. So, the strategy to eliminate floppy mobile termini works here, and this strategy works for many cases. Then express individual domains separately and crystallize separately. What that means? So, suppose you have a big protein like that. Okay. Now, what you do, something we call restricted proteolysis. What is restricted proteolysis? Restricted proteolysis is you get the protein and you add protease from very small to increasing concentration. What will do? The protease will cut your proteins, but with increasing concentration they will start chopping the protein more and more. Now, at different concentration you run the gel and you find that in which concentration of the protease which domain you get, and then you go through purification techniques like size exclusion chromatography, and then you separate the domains and you crystallize them. So, like they have get the biochemistry here, understand them and then get the domains and publish. See, they have done the biochemistry in 1996 and it take them 10 years to get the crystal structure, because this is a very big protein and getting crystal is very difficult. Add a ligand that bridges the domain and locks them together. Sometime what happen, you have two domains and they are like both the domains are stable, but they are moving based on the loop. The loop is floppy. So, the proteins are stable, but they are moving against the loop. In that case, you add a small molecule and you stable the whole system and crystallize it. Like here you see that this is a open conformation, and when the substrate added, this goes to a closed conformation. Sometime you get crystal for both of them and you study the open and closed conformation, you get to know the mechanism how it is working. The fourth strategy is to lessen the entropic penalty to muted high entropy residues, like bigger residues like glue or lysine to alanine. Okay, you have these bigger residues, you mutate them to alanine, or you mutate cysteine to serine when you know that there is no chance of disulfide bonding, then serine will introduce some interaction and it significantly improves the crystallizability. Here in this protein, this is the protein I worked in my PhD, it is called deoxycytidine kinase. The protein was not very crystallizable, but when we change 4 cysteines to serine, we got the crystals. So, this is another strategy. So, by doing that, by taking those personalized strategies, you could significantly improve the chance of crystallization of your protein, and that is what is missing in the structural genomics consortium. So, those personalized strategies would help the small protein crystallography groups. Like if you compare the structural genomics initiative, where there are like thousands of people including big crystallographers, a lot of cloning people, a lot of high throughput things, a lot of money is working, compared to my lab where around 20-25 people are working, 12 PhD students, 2 postdocs, we are much more feeling helpless because we cannot compete with them. But when you look at those personalized strategies, you know that it is difficult to get crystal by high throughput way. So, this is where we get the sunlight of hope, the ray of hope, that even if the structural genomics initiatives are there, still it is possible to do good work with our individualistic, with our personalized initiative, at least when it comes to protein crystallography. Thank you very much. You guys are wonderful audience, and I would end my class here. See you in the next class. Thank you.